Hi viewers, today I am going to explain development of eye in vertebrates. Before going to discuss the development of eye in vertebrates, I would like to discuss some key aspects of development. They are blastulation, gastrulation and neurulation, and these processes are very important for the development of organism. Let us start with the blastulation. Formation of blastula from zygote is called blastulation. Blastula is a single ball-like structure with blastocoele and blastophore. And the cells of this blastoderm have prospective development. The anterior blastodermal cells become ectoderm. The posterior blastodermal cells become endoderm, and cells that are present at blastopore, they become mesoderm. In higher vertebrates, the blastula has primitive node in place of blastopore. So the cells of primitive node develop mesoderm. Let us see how this single layered blastula becomes into three-layered gastrula. Formation of gastrula from blastula is called gastrulation. This single layered blastula becomes into three-layered gastrula. In gastrulation, the cells of blastoderm, they migrate into blastocoele through blastopore or primitive node in higher vertebrates. The movements of the cells here is called morphogenic movements. Let us move to the neurulation. Before going to discuss neurulation, I would like to discuss about notochord, which is very important structure for neurulation. Notochord. This notochord provides a support for axial cytoskeleton in organism. This notochord in higher vertebrates present as nuclei pulposi in between vertebral discs. Let us see the structure of notochord and how this notochord develops in embryo. As I said that primitive groove or primitive node, which is similar to the blastopore, and through which the cells from blastopore, they migrate into blastocoele. And this primitive node develops in at posterior part or the caudal part of the embryo. This primitive node develops at posterior part of the embryo or caudal part of the embryo, and later this primitive node grows towards the anterior side up to the mouth part, and this region is called precaudal plate, and posteriorly it grows up to cloacal membrane, and this precaudal plate becomes into future mouth of embryo, and the cloacal membrane into the future anus. Now let us move to neurulation. Formation of neurula from gastrula is known as neurulation. In neurulation, we are going to see formation of neural tube and neural cage structures. And this picture shows the gastrulation in higher vertebrates. Through primitive streak, the cells of blastoderm enters into blastocoele and forms mesoderm and endoderm of gastrula. And this primitive streak after gastrulation becomes notochord. The notochord is present beneath the outer ectoderm, and the area of ectoderm which has contact with this notochord forms a neural groove structure, and the elevated structures of this neural groove are called neural folds, and these neural folds, they move towards each other and forms a neural tube beneath the outer ectoderm. So the cells that are present in the neural fold, they form neural crust. So after formation of in my previous video, I have discussed blastulation, gastrulation and formation of notochord and neurulation. And in this video, I am going to discuss development of eye. If you have not watched my previous video, please watch the video, and the link for that video I have given in my description. In my previous video, I have discussed blastulation, formation of blastula from zygotic cell, and gastrulation, formation of 3 germ layers from single cellular blastula, and development of notochord from blastophore or primitive node. Then we discuss neurulation. In neurulation, we observed formation of neural tube and a neural crest from ectoderm by notochord. In this video, I would like to focus on the eye development. After neurulation, neural tube and neural crest structures are formed, and let us see the structure of neural tube. This neural tube anteriorly develops into brain and posteriorly develops into spinal cord. At the anterior side, this neural tube also develops sensory organs. This neural tube has a pair of optic grooves or neural grooves on lateral side, and these lateral structures, they grow towards outside and will form optic vesicles. The development of eye starts on 22nd day and it continues up to 8th week of embryo development. Development of optic cup and lens vesicles. Optic vesicle develops from optic groove or neural groove by evagination process. As it goes outside, this optic vesicle makes contact with the outer ectoderm, and the area where optic vesicle makes contact with the outer ectoderm becomes thick, and this thick structure is called lens placard. Then lens placard undergoes invagination and forms lens pit structure, and this lens pit structure brings change in this structure of optic vesicle and forms optic cup structure. Then this optic cup induces change in the structure of lens pit and forms lens vesicle, which dissociates from ectoderm. After formation of optic cup, the lens pit becomes lens vesicle and dissociates from outer ectoderm. Finally, the structures which are required for development of eye, such as optic cup and lens vesicle, they develop from nerve cord and outer ectoderm.